All right, I'm going to get started. So welcome everyone who's here. I'm Gary Rosner. I'm the current chair of the ISBA section on biostatistics and pharmaceutical statistics. And I have great pleasure in introducing our speaker today for today's webinar. Our speaker is Dr. Sebastian Weber, who is Director of Statistical Methods and Consulting at Novartis. He received his PhD in physics from the University of Darmstadt in Germany, and after his first postdoc in soft matter physics, he moved toward the life sciences and completed a second postdoc in systems biology. Both of the positions were at the University of Freiburg in Germany. In 2012, he joined the pharmacometrics group at Novartis Pharma in Basel, Switzerland. He's an expert in leveraging historical control data in clinical trials, designing oncology phase 1 dose escalation trials, and he's involved in pediatric drug development where he applies extrapolation concepts. Sebastian supports the STAN project and as a developer, and his focus areas are on ordinary differential equation solvers and within-chain parallelization. His research interests include the application of pharmacometric principles in statistics, model-based drug development, and the use of Bayesian methods in drug development. And today he's going to talk about his paper entitled Predictively Consistent Prior Effective Sample Sizes. The paper appeared in Biometrics this year, 2020, and his co-authors are Drs. Beat Neuenschwander, Heinz Schmidli, and Anthony O'Hagan. And so, Sebastian, I hand things over to you. Thank you very much for agreeing to give us this webinar.
Well, it's on me to thank you for letting us share our work.
Oh, I'm sorry, one housekeeping thing. If people have questions, please type them into the chat, and then I'll be able to go through them at the end. Sorry, thanks.
No worries. We need to be consistent here. So the following talk, the outline is as follows. I'll first start with an introduction to state the problem and give a motivating example of which shows how we came to this problem. Then I will go through methods for effective sample size and concepts being used in this problem setting. Then I will come to what we have added, the concept of predictive consistency, and explain that in detail. And finally, I will give a summary and an outlook. Okay, so let's start. So the problem statement looks actually very simple, although it turns out it's not quite as simple. So assume we have a statistical model, so some data likelihood P of y conditional on some parameter theta. And we have a prior distribution for that parameter, and it's actually just a one-dimensional problem. We only have a single parameter to worry about. And now, given this problem, we want to now quantify the prior information in terms of an equivalent number of observations of our data, or in a time-to-event setting, this would be equivalent to the number of events our prior is equivalent to. This is also known as the prior effective sample size, and we think it's an intuitive and useful metric to represent an informativeness of our priors. The motivation behind this problem is that soon we have historical control data from previous trials. Knowing now the ESS from these respective historical data as it is represented in an informative prior for some control parameter is certainly helpful to design a future trial. There's another motivation which we discuss in our manuscript, but I'm going to leave it for time reasons out here, which is hierarchical borrowing or analysis for small subgroups. When you conduct such an analysis, it is of interest in the posterior to know the ESS for each subgroup to quantify the amount of information the analysis brings for every subgroup. But really the main problem, how we came to it, is through the use of historical control data for clinical trials. And the example which I will use along is in the indication of ankylosing spondylitis. This is a study conducted by Novartis and reported in 2013 in the Lancet. And this is a double-blinded proof of concept trial to test secukinumab against placebo. In that indication at the time, there was already quite a substantial amount of data collected on the control group. As you can see on the right, a forest plot of the available historical placebo data from 8 different studies. And this historical data was being used in order to formulate an informative so-called Meta-Analytic-Predictive prior, which you see on the left. So this is a prior which was used for the control group. And the endpoint, which I forgot to mention, is a binary endpoint. So it was a responder analysis. The readout was at week 6. So a higher response rate is better. So the trial had a pre-specified success criterion of that one wanted to see that the response rate in the treatment group exceeds for sure the response rate in the placebo group. So for placebo, relatively informative prior was used, and for active and non-informative prior was used, in which you see here. And because the team felt very confident in their prior information, they used a 4-to-1 randomization and conducted the trial with 24 compared to 6 patients. Let's be a bit more specific about the meta-analytic-predictive approach. So this is technically can be described as a hierarchical model to obtain the predictive of a mean parameter, if you want to, also known as random effects meta-analysis. So let Y be the control with summary data from our historical trial. Each and every historical trial gets in this model its own control parameter, pi. So we have as many control parameters as we have trials. And we say that the future trial would actually also, for which we don't have yet collected the data, will also have some control rate pi star, which we are interested in. Now, in order to say something from the past for the future, we link these by an exchangeability assumption on the logit transformed respective parameter space and say that each of these parameters comes from a common normal distribution with a common population mean response beta on the logit scale. And these are allowed to be different, and as such, the between trial heterogeneity tau is greater than 0, and by that we allow for differences between the different trials nonetheless, because they are exchangeable and similar to one another. We learn from past trials for a future trial. So this analysis targets the predictive mean response pi star, which is a predictive mean response for a new trial, and this is used as informative MAP prior. And once you're at that point in the analysis, you of course would like to know, yeah, how much information do I have now in my hand? What's the effective sample size of that parameter? And actually, personally, I got asked this question quite a lot, as I'm also the author of the R package RBEST, which stands for the R Bayesian Evidence Synthesis Tools, available freely on CRAN, which helps statisticians, which is aimed at helping statisticians in the pharmaceutical industry to design map priors in order to leverage historical data and use it for clinical trials. So the tool basically tries to support statisticians in all the necessary steps to do so. So, of course, people will need to collect relevant historical data, which is where the package is not as useful, maybe. But it then helps statisticians to go through all the steps. So synthesize evidence with the map analysis to obtain an informative prior in actually parametric form to be able to write it down into a protocol. Then we typically want to evaluate frequentist design properties for which obviously one needs to know the sample size. And the goal of this whole approach is to be able to reduce the sample size in the control group, given that we can supplement it by the information we have by an informative prior. So what is now the effective sample size of this MAP prior? And even though I almost feel a bit bad sometimes when then answering with an ESS, because it's, when I'm being asked this, it's often in the sense of some alternative hypothesis being tested, and then under some the target power, people want to know the sample size to use. And here I definitely want to emphasize at this point that the ESS is a fully Bayesian metric and has really nothing to do with this sort of sample size, although it's useful to know one to make a decision on the other. RBEST is by now there's going to be a publication in JSS, which is forthcoming. And in this context, I would also like to mention the R package-based Meta from Christian Röver, which since recently, as I know, also supports the ESS ELIA, which we are going to discuss in the following. So methods for effective sample size. Let's look first at the canonical conjugate prior data likelihood pairs, which I would assume is familiar to most of you. So in the left column, we have the data, which is distributed according to some sampling distribution. Then when choosing the respective prior pair with it, we end up having a fully conjugate analysis, and we can solve and update the prior to the posterior in full analytic form. And there are basically three justifications, as I see it, in which way you can justify that the effective sample size of the prior given by its parameters A and B, usually, or N0 for the normal, is actually the formula as you see it. We'll discuss it at the example of the binomial beta. And so the prior to posterior updating rule would be the main and first justification. So if we have now a binomial beta case, so we have data collected for n trials and R responder or successes, then the posterior is given of, for the beta distribution, is given of the form as you see it. So basically the updating rule for the individual parameters is that the prior A becomes A plus the number of responders, and B becomes the prior B becomes B plus the number of failures. And obviously the sum of all failures and successes must be the total sum of all our observations which we have. So you can deem A plus B the effective sample size in this case. Justification 2 is that you can actually write the posterior mean as a weighted average of the prior mean and the sample mean. So in the binomial beta example, the posterior mean is as you see it, and you can rewrite it in this way, and then observe that the non-normalized weights are as follows. So A plus B for the prior and N for the data. So again, it makes sense to say the effective sample size is A plus B. Now these have so far been very specific to the parametrizations and the priors chosen. The third one, the variance ratio, is a bit more general, as it goes away from specific parameters of each prior. And here we look at the ratio of the expected variance from one observation and the prior variance of the mean parameter. And if you calculate it through for the binomial beta example, going through the equations, you find again that the ESS is A plus B. And you can do similar analogy. You can go through these types of justifications for all the other cases on the previous slide. Now let's move to the general case, a non-conjugate case. So maybe you want to use a prior which better reflects your prior beliefs, and for that one, there is no analytical solution possible, or you are interested in historical data, which we synthesize using a hierarchical model for which it is again not conjugate anymore, or it's simply that conjugate prior does not exist in your problem setting. Another question is, what would we now? Another question is now, of course, what to do, how to calculate that. And one requirement which we definitely want to put forward is the ESS conjugacy requirement, as we call it, which means whatever formula or method we come up, if we calculate, apply it to the cases which we just looked at, the generally known and accepted formulas should pop out and come out. So these should then give the same known result. The methods we're going to look at are the following. Basically, these are three things. They build, in essence, on the variance precision ratio justification 3, which we've just seen. So these are variance precision ratio methods. And we're also going to discuss, actually in a bit more detail, the Morita-Tal-Müller method from 2008. And in addition, we're going to discuss the new method, the ESS-ELIA method, which is, ELIA stands for the expected local information ratio, which fulfills a new requirement, which is a predictive consistency requirement, which is not fulfilled by the other methods. Before I start, I need to introduce a few bits of notation. For one, the 1 unit of data Fisher information and the prior information. So the data, in absence of any prior, provides information for one observation, which is the Fisher information, which we write in this form as shown. So it's basically the expectation over the data of the log of the second derivative of the log density, which is, if you will, a measure of curvature. In full analogy, we define the prior information as a second derivative of the log density of the prior. And it's important to note that these are functions of the parameter, not single numbers. So we cannot just simply take the ratio of the two and call it a day and call it that this is the ESS. So that's not possible. Now, the variance precision ratio ESS is relatively straightforward defined. We simply define the ratio as shown here. Both of these give very, very similar results, and I will in the following actually only present the variance ratio method, as they really they are conceptually basically the same. Now, Morita, Tal, and Müller defined or proposed in 2008 another method, which is a bit more complicated, but it was also at the time very ambitious in terms of defining an ESS in a multivariate case. And it turns out that actually this is also a precision ratio type of method, and it can be in one dimension simplified quite a bit, which we'll get to in a moment. The idea of the method is actually quite neat, as I find, is that it specifies the ESS as a result of an algorithm. It says, find the sample size m such that the information of the prior is equal to the expected posterior information. And this expectation is to be done under the prior. And the posterior information here I'm referring to is the posterior of taking a very flat VARC prior and adding then m events from the prior predictive to it. And from that, we calculate the information. And that needs to be similar or as close as possible to the information from the prior. The prior is not specified exactly, other than that the means have to be the same. So formally, the ESS from Morita, Tal, and Müller is the integer m that minimizes the two expressions here at the top. So this is the difference between the prior information evaluated at the mean and between the posterior information of a flattened prior, if you will, to which we add m events from the prior predictive, and we average out over the prior predictive data coming from the prior. You can then equivalently rewrite this, and you will see this trick a few times in the following slides in this form. And all I've done here is actually writing and spelling out the posterior being the product of the prior times the data likelihood. And you can actually drop the data marginal because we take the information of it. The data marginal depends only on the data, but not on any parameter. And here we take the second derivative when taking the information. So that data marginal drops out. So yeah, it's basically finding, it's evaluating the curvature at a certain point. And some comments to the Morita methods. It's an evaluation at the mean, so at a point, a single point of the entire density, which is, according to Morita, necessary in order to fulfill the ESS conjugacy requirement. As said before, the flat or back prior is not unique, and this is actually a minor issue because the respective prior information is anyway very small. We'll see in a moment that the restriction to integers is actually not needed. And Morita gives an algorithm instead of a formula to find the effective sample size. Now, we can simplify Morita's method quite a bit in one dimension by first noting, because the predictive data which we are using here is independent from one another, is that that this expression can be simplified just to m times taking predictive data of just one observation. And doing so then leads us to that we can directly compute the Morita ESS with this expression. If you now go further and replace the mean with the mode, where you would expect the highest curvature, and drop basically the information from the prior and replace the expectation over one predictive data over the prior predictive with basically the one from the sampling likelihoods, you will end up getting the Fisher information. And this leads to the ESS as defined by Pinello and Thompson, which is very similar. And both of these give actually very, very similar ESS results for the problems we look at. Okay. Let's now return and apply these to the anchor-losing spondylitis example for the historical control data. What I have not yet specified is that, or I've said it, but that we take the map prior in parametric form, but the analysis or the model is, of course, can only be solved with MCMC. So on the top left, you see the MCMC result for the map prior pi star, and this we then simply approximate with 1 beta, which you can obviously see is very imprecise and it doesn't do a good fit to the density itself. And the solution to that, which is implemented in our best, is that we actually fit beta mixtures, and you can, it's been proven that if you use a large number of mixtures, you can arbitrarily approximate any density. And you see if you use a 2-component mixture, the density is already very well described. And using 3 components actually doesn't really improve things much further. The reason to use parametric approximations that these can be written easily into a protocol at the design stage, and as such fully and define what actually is our historical information from this analysis. So we want to know the ESS of these mixtures, basically. And if we now apply the so far introduced methods of variance ratio and the Morita method to the three possible priors, you see that for the variance ratio, you get relatively small numbers of like 24, 25. And for Morita, you get pretty large numbers, actually. And I want to stress here that by using beta mixtures for binomial likelihood, this is still fully analytic, and actually also you still can do conjugate updates. It's just a bit more involved by having to go through mixtures, but nonetheless, it's not very far from the original conjugate analysis from my point of view. But still, the ESS Morita has a far greater ESS than the variance ratio method for these mixtures. And this is intuitively due to the focus on a point of our density, of our prior density at the mean, basically. And at the mean, the curvature becomes very high if you add in more mixture components. So basically the peakiness, if you will, drives these large numbers here, which we always thought of that these ESS are very liberal if you take a Morita ESS applied to this problem setting. Now, the ESS using the variance ratio actually does not increase much more when you actually get a more accurate representation of the map prior, which is also a little bit odd. So you have a more accurate representation, but still you don't get more information being accounted for in the ESS measure here. So somehow, whereas the Morita method basically in a way ignores the tails of our distribution, the variance method maybe overemphasizes the tails of the distribution. So we were thinking, are we missing something here at this point? Because we were seeing this type of behavior in many different examples analogously. And this brought us to the predictive consistency concept. And basically, the basic idea behind it of predictive consistency, I mean, an ESS in a Bayesian framework, if you apply it to a prior, it should be no different to if you apply it actually to a posterior. And if you know how prior got to posterior, namely by knowing how much data went into the prior to become the posterior, then these things in an ESS for an ENS functional should be related to one another. And concretely, what we said is what we want to see is that if we have a sample size of M, then the expected posterior ESS must be the sum of the prior ESS and M. So this sounded extremely logical to us, and let's get a bit more into the details. I said the expected posterior ESS, so we take expectation with respect to the prior predictive density. And what you see here in the plot is you see the ESS taken over the, sorry, I'm using as example the three-component beta mixture from the previous slide. And what you're looking at here is the ESS evaluated for the posterior when adding capital YM data. And M denotes the number of events which have been simulated from the prior predictive, and we integrate out according to the prior predictive density. And as such, of course, we get an ESS, which is a lot larger than what is the ESS of the prior. But if we now subtract, again, M, the number of events I have forward simulated, then, of course, I should get back the ESS of the prior. And what I'm plotting now to you here is on the y-axis, this expression, which should actually give you the prior ESS, which shouldn't change in this sense if it would be predictively consistent. And I'm showing it to you for the variance ratio method in red and in blue for the Morita method. And the nice thing is they both converge onto somehow the same value when just going to large enough forward simulated predictive data. And we thought, well, this is basically what we saw as a plot. We didn't at the time, of course, know about ESS Elia, but we definitely thought, well, if they agree with these large predictively consistent, with these large forward simulated sample sizes to some value, we actually want to know that value. And this is what ESS Elia exactly fulfills and does. So only the ESS Elia is predictively consistent, which means that when doing this forward simulation and taking these expectations, the Elia method will always give you the same ESS. And how is now ESS Elia defined? Here it is. So it is simply defined as the ratio of the prior information and the Fisher information, and we take the expectation with respect to the prior. And this is actually extremely similar to Pinello and Thompson's definition, which, if you will, is a plugin version of the ESS Elia plugin version, because instead of integrating over the parameter, we simply plug in the value for the parameter from where the mode is. You can show this, we've calculated in our paper, that it fulfills the conjugacy requirement. So we get back our known formulas. And it also fulfills the predictive consistency property. So it holds that this equation is true for any m. And the proof of doing so is actually quite neat. And let me try to go through it with you now. So assume we have capital YM predictive data of size m, and then the posterior ESS Elia is equal to this expression. So here I've already written out for you that the posterior is product of prior and data likelihood. And again, the data marginal drops out because it doesn't depend on the parameter theta, but only on the data. Now, this thing we want to integrate or average over the prior predictive. So we want to take expectation with respect to the prior predictive distribution for the data capital YM. As a first step, I swap the order of the integration. And then I observe that basically within that bracket, only this term with the derivative depends now on data capital YM. And when you apply it to that term, you see that this immediately, that this is the Fisher information. It's exactly the same. So, or better, it's m times the Fisher information, because the different predictive data is independent from one another. So we are the last line here, and you see immediately the Fisher information cancels out, and what remains is the ESS Elia applied to the prior plus m. So this proves that for any m, the ESS Elia is predictively consistent when sampling forward consistently with the prior we are interested in. So now let's look at what ESS Elia has to say about the ankylosing spondylitis example. In here, we get numbers which are just in between the other almost extremes, the variance ratio and the Morita method. And since these are, on the other hand, these are now for us relatively realistic values, and these are fulfilling the predictive consistency requirements, such that we feel more comfortable using these numbers and calling that this would be the appropriate effective sample size of the prior. Okay, with that, I'm coming to the summary and an outlook. I hopefully have convinced you that the ESS now as an intuitive metric and is particularly useful for sample size determination in clinical trials where you use informative priors. The various similar precision ratio methods which are available so far fulfill the necessary requirement of what we call the ESS conjugacy requirement. So they reduce in the known cases to the commonly known and agreed on formulas. Still, surprisingly, the ESS can differ considerably between these methods, even though they all agree on the canonical cases, which is really due to the case in terms of how they deal with the tail or with the peakiness of the distribution, actually. And this newly put forward expected local information ratio is built such that it is predictively consistent. So doing forward simulation from the prior and averaging over that is always will be consistent using this ESS definition. And this is why we think that also any future work, if it is targeting the ESS, should actually not only fulfill the conjugacy requirement, but also this predictive consistency requirement, which we think is a very Bayesian perspective of things, and we think that this is a very important requirement to fulfill. In the future, some more research certainly is necessary and answer questions like, what is the ESF if the ESS linear does not exist because the integral is not defined? Are there other definitions possibly of the ESS that fulfill the conjugacy and the predictive consistency requirements? Really that we are concerned about these basically requirements on the functional forms. So it could be something else, but nonetheless, so far we have only found a single definition. And certainly what's a broad, still open field is the multivariate case. So when you have an entire vector of parameters, then things get, from my view, relatively messy and not so simple, because it's not anymore clear what you would be aiming or shooting for. Is, do you want the ESS for a given parameter, which is probably not independent from the other parameters, or do you want the ESS of the entire vector, so summarize everything to a single number? Should there maybe be other restrictions put forward, like what about transformations and all that? Does it need to be invariant under transformations? And all these questions still require some work, and I think it would be certainly very valuable if this can be worked out at some point. And with this, I'd like to thank you for your attention, and looking forward to any questions if there are some.
Thank you very, very much, Sebastian. I don't see any questions in the chat box, so if anybody has any, you can just speak up. I'll just jump in then. Have you continued working on this, in particular, the multivariate case, even just going to a bivariate case?
Yeah, well, I mean, basically, I have done and thought about it a bit more. And for example, in the case of a regression problem, one thinking was there to say one could actually maybe solve the problem first if you say the parameters are your sufficient statistic. Maybe then this thing becomes a bit more easier to tackle. But then immediately, if you then want to solve it for a linear model, then you may end up somewhere useful. But if you then want to go to general linear model already, then things get again extremely messy. So, and this paper came out with a discussion, with a few discussion papers of our paper, and there it was put forward to maybe look in more detail onto a multinomial problem, and where you have a Dirichlet prior. I think this was a good suggestion, although we looked at at the time already a bit more into things and didn't yet come to a conclusion. So it's still ongoing, I would say. So, but yeah.
Before I follow up on that, Lucas Widmer wrote that the chat box is disabled, so suggested that people put questions in the Q&A section. So thank you for that. So even the linear regression case is interesting. I mean, considering that one may be interested in in evaluating a treatment effect, in that case, it could be conditional, say, on stratification variables. And so it's back to sort of a single parameter, but you're conditioning on the others. I don't know if that would help. And also for generalized linear models, if you're dealing with exponential families, there are conjugacy kinds of formulas that are available. I don't know, just thinking out loud.
Yeah, and particularly if you think about treatment effect, of course, if you condition away things, then, and basically, I mean, probably one first step could be to condition as much as up to just one parameter, and then you should get the sensible answer from what we have here. And then the question would be how that extends if you open up to 2 parameters. But still, I mean, treatment effect is, again, the difference between 2 groups, but you only collect groups on a given, like on group A or group B. So it's yeah, then it's like, if you know something about group A, it will mean something or have an effect on what the parameter of the treatment effect will be, given what you know about B. So it's a matter and a question certainly also, as I said, about the specific parameterization. And I think one also needs to think about if the ESS given should be invariant to parameterization or not. And I think the answer is probably no. And when all these things, one needs to work out in terms of being aware of them. And I would hope that maybe at some point we have another requirement or so, and concept which will guide the way. But yeah, I don't have the answer yet. But yeah, I mean, these are interesting avenues certainly.
Okay. I don't see any more questions. So that just speaks to the clarity of your presentation.
I hope so.
All right. Well, taking as a guiding principle that no one ever complains about ending a meeting early, I will take this opportunity to thank you once again for presenting this work to us. And again, it was very clear, and I think it's very interesting work. And I also want to take the opportunity to wish everyone happy holidays, and hopefully 2021 will be much better than 2020 was. And that's that. So see everybody in the new year, and take care.
Thank you very much.
All right. Bye-bye.
Bye.